Module 5, Cell Signaling Part 2. So in the first part of our discussion, we looked at cell signaling in general, the general aspects of cell signaling. We looked at hydrophobic signaling molecules, and we ended with a discussion on G-protein coupled receptors that could either interact with stimulatory G-proteins to activate adenylyl cyclase or inhibitory G-protein that could inhibit adenylyl cyclase. So now what I want to do is turn back to the stimulatory G-proteins which activate adenylyl cyclase, which results in an accumulation of cyclic AMP in the intracellular space in the cells and see what happens downstream of cyclic AMP. So on this slide, you can see your G-protein coupled receptor that has interacted with a ligand. The activation of the receptor then will activate the stimulatory G-protein. The alpha subunit of the stimulatory G-protein can activate the adenylyl cyclase, and you can get cyclic AMP production in the cell. The next step involves a protein known as protein kinase A. And sometimes it is referred to as A kinase, sometimes it is just abbreviated as pKa. So the protein kinase A is a fairly large kinase. Remember that kinases are the proteins or enzymes that can phosphorylate other proteins. So pKa contains 2 regulatory subunits and 2 catalytic subunits. When all 4 of these subunits are interacting with each other, the pKa is inactive. So it is an inactive A kinase. However, the cyclic AMP can bind to the regulatory subunit, and as you can see that there is a conformational change such that the catalytic subunit, the actual kinase domains, are released in their active state, and they can go on to phosphorylate other proteins in the cell. So the next step after the cyclic AMP is activation of the protein kinase A. So that has been achieved in this portion. Now you remember in the first part of our discussion on cell signaling, we discovered that the same signaling molecule can cause different things to different cells. And for example, we used acetylcholine, and you can see here reviewing the slide that acetylcholine can cause contraction of the skeletal muscle, it can cause relaxation of the heart muscle, it can also induce secretion in the secretory cells. So really what happens depends on the cell signaling machinery which is inside the cell. So up until now, all cells that are signaling through the G-protein coupled receptors do the same thing. They activate adenylyl cyclase, you have accumulation of cyclic AMP, and then finally activation of protein kinase A or pKa. So what happens downstream of pKa depends on cell types. And we are going to look at 3 examples of what happens downstream of pKa. So in muscle cells, we observed glycogen breakdown. In many different cell types, we can see alteration in transcription. In epithelial cells, we observe the release of fluid or passage of fluid across the epithelial layer. So let's look at each of these individually. So in the skeletal muscle, the major storage for energy is glycogen and can be readily converted into glucose 1-phosphate, and then the glucose 1-phosphate can enter into the glycolytic pathway, and the end result is, of course, ATP. So adrenaline is a signaling molecule, and that can induce a signaling pathway that results in the breakdown of glycogen to glucose 1-phosphate and induce glycolysis and the production of ATP. So that's what is shown in the slide. So here is adrenaline. Adrenaline is also known as epinephrine as well. And adrenaline binds to its receptor. Its receptor is G-protein coupled receptor that can activate the stimulatory G-protein. The alpha subunit of the stimulatory G-protein then activates the adenylyl cyclase, and cyclic AMP then activates the protein kinase A. So the pKa then phosphorylate a phosphorylase kinase. And when phosphorylase kinase is phosphorylated, it becomes activated. So the phosphorylase kinase then in turn phosphorylate glycogen phosphorylase. Glycogen phosphorylase, when it's active, when it's in the phosphorylated form, can convert glycogen to glucose 1-phosphate. So here you can see the skeletal muscle cells have this intercellular machinery. You have a number of these relay proteins such as this G protein stimulatory protein, then you have protein kinase A, then you have phosphorylase kinase, then you have glycogen phosphorylase that are all regulated either by binding to GTP or by phosphorylation. And this pathway should make sense to you. Adrenaline is often produced in a situation of stress, and when an animal in a stressful situation or in an active physiological state, say when you are working out or doing any strenuous activity, the skeletal muscles need ATP to flee from a source of stress or to continue in the active physiological state like running or any kind of activity that use muscles. And so it makes sense that the adrenaline should induce these specific signaling pathway in the muscle cells. So one thing is important in here, that what you are seeing, that the one adrenaline, when binds to a receptor, that can stimulate hundreds of G proteins. So at the very beginning, not only that these will amplify the cascade, but also remember that at the very beginning, one receptor which binds to a single molecule of adrenaline will stimulate many of these heterotrimeric G proteins to activate many adenylyl cyclase, which will produce a lot of CAMPs, which will phosphorylate many inactive phosphorylase kinase, and so on. So at each step, there is a signal amplification, and this response is very, very rapid. All right, so as I mentioned, that in another thing that can happen downstream of PKA are changes in gene expression. And this happens in a number of different cells, and that's shown in this slide. So here at the top of the slide, that you can see a G protein coupled receptor that is bound to its ligand, and this is a stimulatory G protein coupled receptor. The activation of the ligand results eventually in the activation of adenylyl cyclase. Please note that there are a couple of steps that are not shown in the diagram. So for example, the heterotrimeric G protein itself not shown in this step 1. So this accumulation of the CAMP then result in the activation of the protein kinase A. And you can see that another rendition of protein kinase A in this figure with its 2 regulatory subunit and its 2 catalytic subunits. So cyclic AMP binds to the regulatory subunit, resulting in the release of the catalytic subunit of the PKA. The activated PKA then enters the nucleus in the step 3, and it phosphorylates a protein known as CREB, or CAMP response element binding protein. So phosphorylated CREB can bind to cyclic AMP response element in the promoters and enhancer of a wide variety of genes. And the CREB can bind or attract to a protein called CBP P300. So CBP is the CREB binding protein, and P300 is also known as EP300, or sometimes also known as a adenovirus E1A binding protein. So together, the CREB along with the coactivator CBP and P300 form an extremely powerful transcriptional activation complex that can induce the transcription of a wide variety of genes. The following is not needed for your exam, but just to let you know if you are curious, that P300 is a histone acetyltransferase, and it remodels the chromatin to allow the histones to wrap DNA less tightly to allow transcription. So the tight wrapping of the DNA around the histone in the nucleosomes inhibits DNA transcription, and the DNA needs to be loosened up for transcription, and that is achieved by this signaling pathway. So that's the second example of what happens downstream of protein kinase A. So the third example of what can happen in a downstream of protein kinase A is specifically occurs in the epithelial cells. And in this case, the stimulatory G protein signaling pathways are activated, and you can observe the movement of water from the basolateral surface of the epithelia to the apical side. So not shown in the diagram, but you have the binding of some signaling molecule to the cell surface receptor, activation of the stimulatory G protein, activation of adenylyl cyclase, and then finally shown in the slide is the accumulation of the CAMP, then the activation of the PKA, and the final outcome is the flow of water across the epithelia. So how does this flow of water occur? Well, as it turns out that this pathway relies on a plasma membrane protein that we have already looked at when we were looking at the membrane proteins. And that's the cystic fibrosis transmembrane conductance regulator, or CFTR. You will remember that CFTR is a member of the ABC superfamily of transporters. Just to remind you, although it uses ATP, it is not a pump but a channel because it transports chloride down the concentration gradient like a channel, not up the concentration gradient like a pump. A typical characteristic of all ABC superfamily of transporters, there are two transmembrane regions and there are two ATP binding regions. And you will remember that the CFTR transmembrane protein is found in pretty much every pathway that is lined with the epithelial cells in our body, like lungs, liver, pancreas, intestine, reproductive tract, and there is even CFTR in skin cells, in skin epithelial cells. In the boxed region of the slide, there is some more detailed rendition of the CFTR, and the yellow are the transmembrane regions. Sometimes they are referred to as membrane spanning domains, MSD1 and MSD2. And then in gray, you have two ATP binding regions. These are also known as NBD1 and NBD2, which stands for nucleotide binding domain 1 and domain 2. And then there is a very important region which we didn't talk about, is the R region, or the regulatory region. So this R domain that really plays an important role in turning this transporter on and off, which is related to the flow of water across epithelia after signaling. So let's see that how this actually works. What happened is a G protein coupled receptor binds to its ligand and becomes activated. Then the stimulatory G protein in turn leads to the activation of the adenylyl cyclase. One then sees the accumulation of cyclic AMP and then the activation of the protein kinase A that can phosphorylate the R domain of the CFTR. And when the R domain is phosphorylated, the transporter can become activated. And we already know what this transporter does. Once CFTR is activated, it transports chloride ion from the intracellular space into the mucus, and it is secreted by these epithelial cells. In this case, it is the airway lumen. The net negative charge then attracts sodium ion to move from the basolateral side of the epithelium to the apical side. So now you have sodium chloride in the mucus. And so now you have this salt gradient, which then draws water across these epithelial cells. And so in this way, the G protein coupled receptor that was interacting with a stimulatory G protein can then induce the flow of water across the epithelium. And you will remember from our discussion on the membrane proteins that the CFTR transporter is in fact defective in the individuals who have cystic fibrosis. No chloride is transported out in response to signaling, and therefore there is no movement of sodium and no movement of water across the epithelium. And you end up with a mucus layer that is quite thick. In the case of airway, mucus binds to pathogens, and individuals with cystic fibrosis are prone to respiratory tract infections and also difficulty in breathing. So when we were talking about the cystic fibrosis, we talked about the types of mutations that you see in the CFTR. And this is very interesting. There are about 800 different mutations that have been associated with CFTR. But the most common in about 70% of the cases is a deletion of the phenylalanine at the position 508. And when you delete the phenylalanine at position 508, you end up with the misfolded CFTR while it's being synthesized in the ER. And this induces the unfolded protein response. And you will remember from our discussion on vesicle trafficking that when a protein is misfolded in the ER, it is transported into the cytosol and is degraded. So most of the individuals who have this particular mutation, they are missing this CFTR transporter altogether because it is degraded through the unfolded protein response. So when we talked a little bit about the genetics of the cystic fibrosis, we mentioned that it was autosomal recessive. This is a very simple slide reminding you what autosomal recessive is. If you have a father that is a carrier and a mother that is a carrier, then you have a chances of having a child that has both mutated forms of CFTR genes are 1 in 4. And you must be homozygous for the mutation before you can have cystic fibrosis. So the heterozygous individuals are not affected and do not show any severe symptoms. And of course, there will be 1/4 which will not be affected at all. And here is what the numbers look like. And you remember this also from our discussion on membrane proteins. Again, it's a recessive trait. It affects mainly Caucasians. 1 in 2,500 children are born with this disease. About 30,000 people live with cystic fibrosis in the US. And this is very interesting. About 1 in 20 individuals are carriers. And we didn't elaborate on this point, but I want to elaborate on this now because the point that 1 in 20 individuals are carrier is highly related to our discussion on cell signaling. Okay, so this frequency of 1 in 20 is very, very high and is much higher than you would expect given the deleterious effect of having mutated or non-existent CFTR. So why is it so prevalent? And several years ago, some scientists asked the question of whether the CF gene confers a selective advantage to heterozygotes. So is there some kind of selective advantage to the individual who are heterozygous to the CFTR mutation for the prominent one, which is the deletion of phenylalanine, or even any other mutations? And this is similar to the sickle cell anemia story that I think many of you probably know the story. Individuals who have sickle cell anemia tend to be resistant to malaria. And resistant may be kind of a strong term, but that's why I have them in the quotations. They can get malaria infections, but they survive malaria infection, so they are less sensitive to the malaria infection. So it is well known that there is an increased incidence of sickle cell anemia in individuals of African descent. And the idea is that perhaps the malaria in subtropical and tropical regions of Africa selected for the individuals who have sickle cell anemia. So in other words, here is an infectious disease in which you have an unequal survival of individuals, and the individuals who have a certain trait, a certain genetic mutation. So in this example, individual with sickle cell anemia actually survive infection, so their rates in the populations are higher than the expected. So this question is, did that same thing happen for cystic fibrosis? Was there some kind of selective advantage for heterozygotes? Why are so many heterozygotes? Why are 1 in 20 individuals are carriers, and so on? And so you have to ask the questions, are there any infectious disease that might affect CFTR channel? Or more specifically, are there any infectious disease that might influence signaling from stimulatory G proteins, which in turn would influence the function or activation of the CFTR channel? And in fact, we talked about this in the first part, and that's cholera. So cholera toxin can affect stimulatory G proteins. So let me remind you that this is the slide from the part 1 of the cell signaling talk, and you can see that the cholera toxin can covalently modify that alpha subunit of the stimulatory G protein, locking it in a GTP bound state, and it is perpetually on. It cannot be unactivated or deactivated. So let me just remind you that the cholera is a diarrheal disease. It is caused by the bacterium Vibrio cholera. It is a waterborne infection, and one of the most prominent symptoms of cholera is the secretion of chloride and fluids into the intestine, resulting in very watery diarrhea. And remember that CFTR mediates the movement of fluid across epithelia. So is there a connection? So let's look at how cholera works, and then let's look at how cholera may have selected for individuals who are heterozygous for the cystic fibrosis gene. Okay, so cholera toxin locks the stimulatory G protein in the GTP bound state or on state. The result is a significant increase in the amount of cyclic AMP, so much higher than a normal. This in turn results in significant activation of the protein kinase A. So again, much higher than normal, which in turn will result in significant overactivation of CFTR. And in this case, you have a significant amount of chloride being released from the cells and then followed by sodium, then followed by water. So you have a very large volume of water. You have a very severe diarrhea. So why does the pathogen want to do this? The mechanism of action, as you can imagine, is to facilitate the spread of the pathogen to a new host. So diarrhea, or this discharge of the stool, and which goes into the environment, that is basically the mode of transmission of the pathogen. Okay, so did cholera select the individuals with CFTR mutations? And this was the approach that the scientists took. What they did, that they knocked out the CFTR gene in a mouse model. They then bred those mice to get both homozygous and heterozygous offsprings. And then they exposed the mice to cholera toxin. And then they looked at what happened to the mice, how sensitive they were to cholera toxin, and what happened to those intestinal cells. So here is the data from the experiment. So you can see in the bottom on the x-axis, here to the right, you have the individuals who are plus plus. They have the homozygous wild type CFTR. Then in the center bar here, they are heterozygous for the CFTR. And on the left-hand side, you have the individual mice who don't have CFTR transporter at all, minus. And then these mice were exposed to cholera toxin, and the scientists measured the accumulation of fluid in the intestine. And you can see the individuals who are wild type has a very significant accumulation of fluid in the intestine. And the individual who are heterozygous or homozygous for the CFTR mutation or knockout of CFTR secreted mucus, much less fluid. And in fact, that these individuals, the heterozygous and the homozygous, had much milder infection and greater survivability. And so this suggests that cholera, and specifically cholera toxin, selected the individuals who maybe are heterozygous or homozygous for cystic fibrosis gene. And this might have increased their number in the population. So we also talked about another pathogen, that is Bordetella pertussis, which is the causative agent of whooping cough. And what it does is the pertussis toxin can bind and modify the alpha subunit of the inhibitory G protein. So the question is, did the pertussis toxin also help in the selection of the cystic fibrosis gene or mutation in that gene? Okay, so now here, what happens in Bordetella pertussis infection? So now we are talking about the lumen of the respiratory tract. So Bordetella pertussis produces a toxin that locks the inhibitory G protein in its off state. So it is a GDP bound state. The outcome of that is a significant increase in the cyclic AMP. This is the same outcome that we saw in the cholera infections. So just take a moment to think about this. If you cannot turn on the GI, heterotrimeric G protein, if you cannot turn it on, then you cannot turn off the adenylyl cyclase, because it is the inhibitory G protein that turns off or inhibits the action of adenylyl cyclase. So the outcome is increased intracellular concentration of the cyclic AMP, which in turn causes increased activation of protein kinase A and increased activity of CFTR, and then of course, large volume of water across the epithelial layer into the respiratory tract, producing characteristic whooping cough. It is almost the patients are drowning. Individuals with the disease almost sound like that they are drowning, and like for cholera, this actually facilitates the spread of the pathogen because it induces coughing in the host. Now, is it possible that pertussis also selected for individuals who have CFTR mutation or cystic fibrosis? It is not really known. The frequency of whooping cough in CF patient is not known. Also, widespread whooping cough outbreaks have not occurred like those for cholera, so I don't think an experiment like cholera has been done, but you can think about that how you would do this experiment. Just a note that there has been an upswing of the whooping cough for the past 10 years. It is thought due to the mutation of the bacterium cell surface, which makes the vaccine not as effective. This concludes part 2 of Module 5 Cell Signaling discussion.